Well, here we are at video 3 on photosynthesis, and we've got one more video that we're going to be looking at, but that'll be a short one. I just thought I'd show you first of all my new super duper microphone that I'm using. It looks like something from, I don't know, the 1950s or something. I've borrowed it from Mr. Spears. I thought maybe to liven up some of the videos in the future, since you're busy listening to me, I might actually get some fancy wigs or something to liven these up, or try and get somebody else to record them so you can listen to a different voice. But meanwhile, you've got me. So on this video, we're going to be looking at photosynthesis and what are known as limiting factors of photosynthesis. Now, to look at limiting factors, what we need to do is think about, well, what does photosynthesis need to work, or what things can photosynthesis, if they're missing, the photosynthesis cannot work without them. So we've got carbon dioxide there, that's needed for photosynthesis. We also need to have sunlight. And as well as that, remember I said in the first video that the photosynthesis process is controlled by enzymes. So enzymes are affected by temperatures. So you need to have the right temperature for the enzymes to work. And these are all limiting factors. If any of them are in short supply, then they will limit the rate of photosynthesis. And by limiting the rate of photosynthesis, we mean we will slow it down. So you need to know that the three limiting factors are temperature, carbon dioxide concentration, and light intensity. And the whole of this area all revolves around this and looks at measuring the rate of photosynthesis and looking at these different factors. So there are various investigations that take place, and there's various exam questions that come up all round about this thing, looking at this idea of these limiting factors. And to measure the rate of photosynthesis, remember we talked in our first video about measuring how much starch is produced. That is one way of measuring the rate of photosynthesis, but it's quite tricky to actually measure that easily. So we could do something like measuring the rate of carbon dioxide uptake, how quickly that happens, and to do that we need to be using something like a carbon dioxide probe. So again, that's quite a tricky thing to do. So one of the easiest ways to measure the rate of photosynthesis is to measure the amount of oxygen that's produced in a plant. Now that can be used, I mentioned in the first video, I think it was, that we could use an oxygen probe to do that, but there's another way which is really quite easy and simple and straightforward, and that is to measure how much oxygen is produced by a plant which lives in water. And these are the two plants that you kind of come across in photosynthesis and limiting factors questions. One of them is called kabomba, and you have used kabomba in the classroom already. And the other one is a very similar water plant, and it's called elodea. And what happens is that when these plants are photosynthesizing, they produce little bubbles of oxygen, and these little bubbles of oxygen can be counted. And if we can count them over a period of time, like say a minute, then we can record how many bubbles of oxygen are produced per minute, and it gives us an idea of the rate of photosynthesis. The faster the bubbles are produced, the faster the rate of photosynthesis. The slower the bubbles produced, the slower the rate of photosynthesis, and we can get measurements of this. Now, the sort of experiments that can be done, we'll have a wee look at the kind of experimental setup, because these come up in exam questions sometimes as well. Now, this is a very simple way of measuring the rate of photosynthesis. And in this experiment, we've just got a plant here. In this case, in this diagram, it's actually elodea, and it's in this test tube here with a solution that's got bicarbonate in it. That bicarbonate gives the plant some carbon dioxide. Now, in this experiment here, the lamp is providing light to the plant, and we can move the lamp backwards or forwards, and that will change the light intensity. So we could measure, say, the light intensity at this point here, say it's maybe, I don't know, 5 centimeters away from the plant, count the number of oxygen bubbles given off in a minute, and that will tell us the rate of photosynthesis. Then we can move the lamp further away and say 10 centimeters away, and then we can count the number of bubbles. Now, there is an experimental error with this here, because we're changing the light intensity in this experiment, And one of the problems is, remember that if you're changing one variable, you have to keep all the others the same. So we're keeping our carbon dioxide concentration the same because we will use a certain concentration of bicarbonate. But the problem is here we've got a lamp that's quite near to the plant, and so that could be giving off some heat as well and may affect the results. And sometimes in exam questions it might ask you about that. And you can solve that by just putting maybe a glass screen up here between the lamp and the plant, and that prevents the heat affecting the results. Now, a more complex setup would be this kind of setup here, where you've got a plant here trapped below a kind of funnel. I'm saying it's complex, it's quite straightforward to do, but you've got this funnel here and you put a test tube over this. So you've got your indicator, your bicarbonate, it's not indicator, sorry, you've got your bicarbonate solution here, giving carbon dioxide. You could change the light intensity or the bicarbonate solution, and you could count again the number of bubbles, you could count them, or you're trapping the oxygen up here, so you could actually measure. If this was a measuring cylinder, for example, you could actually measure the volume of oxygen that's been produced here. And if we just have a look at this next wee diagram, it's got the same kind of setup here, where you've got this upside down test tube, you've got the LED or plant here, and in this case here we've got a thermometer. So one of the things that could be done, you could change the temperature of the solution that the plant is in, and you could measure the rate of photosynthesis as long as you kept the lamp at the same place and you kept the bicarbonate, or in this case here it's called sodium carbonate, it's just providing a source of carbon dioxide, as long as you kept them the same. Or say you decide that you want to investigate the effect of light intensity. Again, like in the example that we had before, you could move this lamp back and forward. Having this set up here, again, there's not a glass screen here, and you could put that up to prevent any heat from the lamp affecting the results. Or the other thing you could do is you could keep the temperature the same, you could keep the light intensity the same, but you could change the sodium carbonate here. You could make the solution stronger. You could maybe look at a 5% solution, a 1% solution, a 0.1% solution, and look at the effect of changing the carbon dioxide concentration in your experiment. Now, when you've set up an experiment or an investigation like, you will get a graph from it, and because you've got a set of results, and you would graph it out. Now, I think I won't use this graph here, sorry. I'm going to move on to this other one here. And that is, if we look at this graph, I've got a lot of writing on it just now, but let's just look at the axis of the graph first of all. So here, down here, we've got light intensity. So it could be that what you've got is you have, if we just see here, this is the nearest, or the furthest away point actually would be a better way of putting it, from the plant. So you've got 0 light intensity. And then as you start to get nearer the plant, in other words, you're increasing the light intensity. So as you start to move your lamp nearer the plant, the rate of photosynthesis will start to increase. Now here, up the side here, is the rate of photosynthesis, and that could be something like measuring how many bubbles of oxygen are produced per minute. So you've counted that. So you count the number of bubbles down here, and possibly there's been none, and then maybe you moved the lamp 10 centimetres nearer, and so you maybe have counted, I don't know, 20 bubbles in a minute, and so on. And so you would start to get your results and plot out your graph, and you'll get this increasing slope coming up like this. Now eventually you reach this point here where after that the graph stays level. In other words, the numbers of bubbles of oxygen that are being produced, it just stays the same no matter how much more light you give the plant. You can move it nearer and nearer and nearer, but it doesn't actually change the rate of the photosynthesis. And the reason for that is because now it's no longer light intensity that's limiting photosynthesis. There's something else that is limiting it. It could be, we've got our other two factors, it could be your carbon dioxide concentration, or it could be the temperature. So it doesn't matter whether you give that plant, a whole sun to give it more light. Something else is limiting the photosynthesis. So this part here will just always remain at this level unless you change the carbon dioxide concentration or unless you change the temperature. So all your exam questions all round about this are based on graphs that actually look like this. This is the basis of the kind of graph. You'll have some sort of factor that's down at the bottom on the x-axis. And as a rule, as I've said here, whatever is on the x-axis is the limiting factor on the slope part of the graph. So what we're saying here is that from this point here to this point here, whatever is down here on the x-axis is what is limiting photosynthesis. So at this point, it's light intensity, because that is what is down here. But when you come to after this point here, something else is a limiting factor, and that could either be carbon dioxide concentration or it could be temperature. So let's just have a wee bit more of a look at these graphs without quite so much writing on them. And if I just move on to the next slide, here's another one here. Now this graph here, you'll see that we've got carbon dioxide concentration down here at the bottom. And again, we've got the rate of photosynthesis, and that, as I've said, could be number of bubbles per minute produced, or it could be volume of oxygen, because it might have been that you've actually measured that volume of oxygen in a measuring cylinder produced per minute, so it might be cubic centimetres produced per minute. But anyway, you've got this graph again that's been plotted out, and it's got the same kind of setup as the graph we had before. Now, all the exam questions will be based around this idea of what is limiting photosynthesis at this point here on the graph, or what is limiting photosynthesis along this point here on the graph. And the rule, remember, that we've said is that whatever is down on the x-axis down here is what is limiting photosynthesis at this point here on the slope. So if the question said, what is limiting photosynthesis at point A, then the answer to that would be carbon dioxide concentration. If, though, the question was asking what is limiting photosynthesis at point B on the graph, then the answer to that would be either light intensity or the temperature, something other than the carbon dioxide concentration. And in a similar fashion, if we have a wee look at this graph here, this is one here where we're looking at temperature between nothing and 40 degrees Celsius, rate of photosynthesis again. At point A here, well, we're on this part of the slope of the graph, so the limiting factor is temperature. At point B, we're at this point here on the graph, so the limiting factor is something else. It's either carbon dioxide concentration or it's light intensity. There's a little bit that I'm going to say, though, extra just amongst this, in case a graph comes up that looks slightly different from this one, that is to do with temperature. And that is based on this, well, I've got here what happens after 40 degrees Celsius. Now what you have to do is remember that photosynthesis is something that is dependent on enzymes. So sometimes you might actually see a graph like this, where as the temperature is increased, the rate of photosynthesis does increase, but you don't get this sort of leveling off here. So we're talking here maybe about some temperature above 45, 50 degrees, 60 degrees and so on. Well, think back to your enzymes topic. What is happening up here at these higher levels? Photosynthesis is dependent on enzymes, and what happens is the enzymes for photosynthesis end up being denatured, and so the rate of photosynthesis just plummets, it drops dramatically. So if we have a little recap with some of these limiting factors graphs, here, this graph that is over here, you'll see light intensity is the limiting factor on this part of the slope here. After that, as it evens off, then it will be something else, carbon dioxide concentration or temperature. This graph here, it says that carbon dioxide concentration is down here on the x-axis, so as you increase the carbon dioxide concentration, the rate of photosynthesis starts to increase, indicating that carbon dioxide is the limiting factor here. In other words, as you're giving more carbon dioxide, you get more photosynthesis happening. But then again, it starts to even off, and that is because at this point here, you can give it as much carbon dioxide as you want. It won't make any difference because something else is limiting the temperature or the light intensity. And this here, you could get a temperature graph that looks very similar to the ones that we've already been looking at. In other words, it looks like this and then like this. And you would say that temperature is limiting at this point here in the slope. And after here, it's carbon dioxide concentration or it's light intensity. Or you may see a graph that looks like this one here. And that one there will really be looking at you talking about or thinking about the fact that enzymes are involved in photosynthesis, and that as the temperature increases at higher temperature, the enzymes end up being denatured. Now I want to have a look at one other type of graph that sometimes appears in questions, and it's this here where you've got, I'm just going to get my wee lines here. You've got two lines here. You've got this slope here, and then it splits into this line here, which is Z, and this line here that is Y. And you'll notice that there's light intensity down here, so this is increasing light intensity. You're getting the lamp nearer to the plant, or you're using filters on it, and the filters are letting more and more light through. But as well as that, you'll see that here and here, there are two other pieces of information. Now this tells you here that there's a 0.5 concentration of carbon dioxide and the temperature is at 15 degrees Celsius. This one here tells you it's 0.5 concentration of carbon dioxide and it's 25 degrees Celsius. Now you may get asked then different exam questions round about a graph that looks like this. So you may be asked a simple straightforward question, which is, well, what is limiting the rate of photosynthesis at point X? And that's straightforward because this is the slopey part of the graph, and therefore it's light intensity. It's the thing that is down on the x-axis. But then it asks the question, what is the limiting the rate of photosynthesis at point Z? And we've said before that when it starts to level off here, it could be carbon dioxide concentration, or it could be temperature that is limiting it. But we can't say for definite which one it is. Now, in order for us to be able to do that, we need added information, and they've given us added information here. So if we look at point Z here and at point Y here, you will see that there's the same concentration of carbon dioxide being given to both of these. But the temperature here and the temperature here are different. So that is telling, if we have a look at our results, that is telling us as we come up this slope here and this starts to level off, well, what is limiting the photosynthesis at this stage? Is it the carbon dioxide concentration? Well, it's not that, because when we look up here, the carbon dioxide concentration is the same up here. So there's not been any change made to the carbon dioxide concentration, but there has been a change made to the temperature. And what we're doing is we're saying, well, at 15 degrees Celsius, temperature is limiting the rate of photosynthesis here, because if we give the plant a higher temperature, photosynthesis will still keep occurring. So we can say here for certain that what is limiting the rate of photosynthesis is the temperature, because if we give it more heat, then we get more photosynthesis. But at point Y here, we have now started, this has now started to level off, So what is limiting photosynthesis at this point here, at point Y up here? Well, again, that could be the carbon dioxide concentration, because we haven't changed that at all, so it could be that. But it could also actually be temperature as well, because we could maybe try giving this even more temperature and the rate of photosynthesis will increase, but the chances are that it's probably carbon dioxide concentration. So it was just to let you see a graph like that. I haven't seen one come up so far in the National 5, but I've seen them in the past before at Intermediate 2 level, so I just wanted to show you that kind of thing. So it's a matter of kind of looking at your graph and eliminating what has been changed and what has been given and so on. Now what we'll do is we'll just have a look actually at a past paper exam question round about this kind of thing and limiting factors. And here we've got this graph here which shows the rate of how the rate of photosynthesis is affected by the concentration of carbon dioxide. So you'll see down here we've got a low carbon dioxide concentration and then increasing here to a high one. And then here we've got a rate of photosynthesis in some sort of units, bubbles per minute or cubic centimeters per minute or whatever. Now the question says state 2 environmental factors which could limit the rate of photosynthesis between points P and between points Q. Now it might panic you a little bit when you see this here. Oh, whoopsie daisy. When you see there's 2 environmental factors, because you might say, oh help, I don't remember doing environmental factors round about limiting factors in photosynthesis. What the question is actually just asking what you have been looking at already. What are the 2 things that are limiting photosynthesis up at this point? So you look down here and you can see that carbon dioxide concentration is a limiting factor on the sloping part of the graph. So therefore 2 things that are limiting photosynthesis here will either be your light intensity or your temperature. And that gives you the marks for the question. Now, I would just like to highlight here that they won't accept you just saying light and heat. You need to say the words light intensity and you need to say temperature. They won't accept heat for this question. But you can see how it's quite simple and it's quite straightforward to answer a question like this. And we'll have some practice with this in class. And that is the end of limiting factors in photosynthesis. I hope you enjoyed.